Good afternoon. Good afternoon. Are we good? We good now? Okay. Just some general business announcements. Remember, you have an exam when? Monday. All right. So your review sheet, study guide will be posted by Friday morning. Okay. Everything that we cover through class on Friday is or can be on the exam. Okay. So we have quite a bit to go through today. As we lead up to the exam, we left off on today's Wednesday, Monday, going over what? We finished going over ATP and basic molecules. All right. So the next, we're leading into macromolecules, all right, which is where we're going to talk about today. Macromolecules that we are concerned with: lipids, carbohydrates, proteins, nucleic acids. Okay? So, for carbohydrates is where we're going to start, and for these macromolecules in general, understand that they are big polymers made from smaller subunits. Okay? So for each of these, carbohydrates, lipids, nucleic acids, and proteins, understand what the subunits are, what little parts make up these bigger parts, these bigger molecules, and know how they are made. All right, so that's sort of the preamble of this is what you should know. Something in my eye there. Wink, wink. Okay, get it? Okay, good. I don't have to spell it out for you anymore. So, a polymer. All right, and what these macromolecules are polymers. All right, so it's a long molecule consisting of many smaller building blocks, similar structure, and so on. All right, so we call the smaller building blocks monomers. Multiple monomers put together yield a polymer. Okay. Some molecules that are monomers also function on their own. Okay, you'll see in carbohydrates, glucose is considered a monomer of more complex carbohydrates. Glucose by itself has its own function, right, as an example. All right, how do we synthesize and eventually break things down? Okay, so the buildup of polymers is a certain chemical reaction. You have to take two monomers together and form a chemical bond between them in order to produce a polymer. Like, that's the concept. Okay? We do this by a reaction called a dehydration synthesis reaction. So we're going to synthesize something new from smaller parts. That's the polymer, whatever it might be. And to do that, dehydration refers to or means what to you? What's going to happen? I'm going to take water out. All right? So a water molecule is going to be lost, and in its place, a chemical bond will be formed. Right? The opposite reaction to that, okay, is a hydrolysis reaction. What do you think is going to occur in a hydrolysis reaction? You're going to add a water molecule, and you're going to break a polymer or a larger molecule into its smaller subunits. All right? And these are facilitated by enzymes, which speed up chemical reactions. Okay, so let's draw out what a dehydration synthesis reaction will look like. So here's a glucose molecule. Here's an OH that comes off of it. I'm going to add that to another glucose molecule with OH over here. Okay? What we're going to get is, here's your water, right? So water comes out, and you end up with your two glucose molecules linked together through what we call a glycosidic linkage. It's not the best drawing in the world, but you get the idea, right? Water, that OH from one monomer and a hydrogen that's off the hydroxyl group of another monomer, they come out. And now that oxygen that's left, that's right here, the oxygen that's left is looking to satisfy that outer valence shell. It does that. Now there's also a carbon that's right here that's looking to share electrons. Hey, you want to share? I want to share. Sure. Let's form a bond. That's where the glycosidic linkage is formed, giving you what this is called a disaccharide or a polymer. Questions on dehydration synthesis? I will learn how to talk today. All right, here it is again. All right, so you have a short polymer. Now, subsequently, if I wanted to add another glucose to what we just saw, I would have to do another dehydration synthesis reaction at the other end. Okay, so that's what they're showing here. You have a polymer, all right? So you have the three monomers that have been brought together with a hydrogen on one end and an OH on the other end. Okay, here's your OH. Your H2O is going to come out, and that polymer now is going to be added to the chain. Okay, on the flip side, hydrolysis. To take one of those off, we have to put water in, which then hydrolyzes this bond and separates one of the monomers off. Okay, so one takes water out to form a bond, the other puts water in to break a bond. Questions on dehydration synthesis or hydrolysis reactions? Yeah.
Is that always like a hydrogen and hydroxyl atom?
It is not always a hydrogen and hydroxyl atom. In this case here, it is because of certain types of bonds that are formed. Okay, so you go through the glycosidic linkage through glucose. There's two OHs, and one of the H comes off of that. So it's not always a hydrogen and a hydroxyl, but they will always produce water in some way, right, to synthesize something. All right. Now, polymers. What are they made up of, or how are they made? Okay. So we're going to talk about macromolecules, all right, that vary among different cells of an organism. Right? Macromolecules, proteins, carbohydrates, nucleic acids, and lipids. Depending on the cell, depending on the species, different macromolecules are utilized. All right? Cellulose is not utilized in animal cells as it is in plant cells, as an example. Okay? So you can, because of adding the way monomers are put together, yields a difference in their structure, difference in their properties, and therefore cells utilize them differently. All right, so that allows for a large variety of polymers and their functions. All right, so first we're going to talk about carbohydrates. All right, function of carbohydrates, or what are carbohydrates? They include sugars and polymers of sugars. I gave you an example of a sugar, glucose. Okay, that is a monosaccharide. Okay, monosaccharide. Or simple sugars. Examples or names: glucose, fructose, and galactose are simple sugars. Okay, are monosaccharides. Simplest carbohydrates, they range between 3 to 7 carbons. Those monosaccharides I just gave you are C6H12O6. Right, they are the 6 carbon sugars. Right, they share the same formula, chemical formula, but they have different structure. Okay? And then you build those together to form 2 sugars, or a disaccharide. All right, glucose plus a glucose is maltose. Glucose plus fructose is very sweet, sucrose, or table sugar. And galactose plus a glucose is lactose, which some of us don't like very much. Okay? So beyond that, now beyond a disaccharide, you can continue to add subunits of glucose to these molecules. And eventually you'll get, depending upon the structure of those glucose molecules, you may get cellulose, chitin, glycogen, or starch. Those are polysaccharides. So starch, glycogen, cellulose, and chitin. All right, so but those are the polymers, and we're going to go through those throughout this lecture today. All right, so here are your monosaccharides. Glucose, C6H12O6. All right, and monosaccharides are classified by number of carbons. All right, and the placement of the carbonyl group as the C double bond O. All right, so those monosaccharides I gave you are all 6-carbon sugars, or hexoses, 6-carbon sugars. All right, you have a pentose, such as ribose or deoxyribose. Those are also monosaccharides. All right, there are 5-carbon sugars. You have a triose, such as glycerol, all right, which is another simple carbohydrate, monosaccharide. Right? But glucose is the most common, the most well-known, and probably the most utilized. Okay, so here's a triose, a glyceraldehyde, and in each of these you see the C double bond O. The difference being between glucose and fructose, same chemical structure, same chemical formula, C6H12O6. Placement of the C double bond O is different. The structure is different. Different structure, different function. Okay? You have a triose, a 3-carbon sugar, one of the glyceraldehyde. Okay? Ribose, a 5-carbon sugar, and then your hexoses. Questions on the difference between glucose and fructose? You should be able to notice, you should be able to identify that difference. Okay? Now, they're often drawn as linear structures. I already drew them as 6-carbon rings. So you are familiar with what they look like. All right? So when I draw something like this, wow, that's awful. But it's a hexagon, to say the least. That is glucose. All right, I'm not going to add the OHs. I'm not going to put in the double bond O. You'll know that that's glucose. It's a 6-carbon sugar. It's different from when I draw, a 5-carbon ribose. All right, and I'm drawing a nucleotide with a PO4 in the end and then A. All right, that's different from 6-carbon sugars. All right, these monosaccharides are used as fuel in the cell. Right? You know that glucose is the energy for the preferred energy substrate. Okay, that's your preferred energy source of for cells. Can cells use other things besides glucose? Yes or no? Yes. Okay, but the preferential and the most bang for their buck comes from glucose. All right, so they get the most ATP out of starting a glucose as a starting material. All right. The other purpose for these carbohydrates are building molecules. They are used for structural support. Some cells use them for identification molecules between cells. That cellulose and polysaccharides are used for structure for plants. Okay, here's the difference between your linear and your ring forms. I'm not going to. Here's your abbreviated ring structure, and which equals to me that. Okay? Keep it simple. All right? I'm not going to draw out. This isn't organic chemistry, so don't worry about that. All right? It's a simple ring structure. I'm talking about glucose. So your disaccharides are formed when a dehydration reaction joins two monosaccharides, forming what's called a glycosidic linkage. Okay? So that linkage between the O, right, between the carbon, this is a glycosidic linkage. All right? Now, difference between this linkage, we can talk about where on how these two glucose molecules are linked together. Some are, this glycosidic linkage can be a 1,4 glycosidic linkage. Some of them can be a 1,6 glycosidic linkage. What do you think the difference is, 1,4 to 1,6? What is the 4 and the 6? What do you have 6 of in these subunits? Carbons. Okay? So what they're numbering, 1,4, 1,6, is how the linkage is between this number, the carbon on one side and the carbon on the other side of the linkage. Depending upon which carbon it is, number 4, number 6 on the second glucose molecule changes the structure. All right? It changes the structure of the disaccharide. Oh, we're not good. All right, so here's your glucose and fructose dehydration synthesis. All right, what are you going to end up with? Again, fructose is a different structure, has a different ring structure. 1, 2, 3, 4, 5, 6. Okay, this is a 1,5 glycosidic linkage. Okay, and that's with fructose, not with glucose. So you end up with your, well, in this case, what we call a 1,2. I numbered from the wrong side. I'm sorry. This is a 1,2 glycosidic linkage. 1 on glucose, the 2 on the fructose, giving you a 1,2 glycosidic linkage. All right? This comes in this becomes very important when we talk about glucose molecules linked together. Okay? Glucose molecules can be linked 1,4. They can be linked 1,6. They can be linked 1,4 opposite, or we call cis or trans. So they're flipped. Okay? Because of the difference in structure, it's glucose to glucose. Big deal, right? But because of the difference in structure, it gives a difference in function and how they're used and recognized. Okay, so polysaccharides, the polymers of sugars, have storage and structural roles. Storage, yes.
I don't know. Can I get a little bit more clarification on the 1 to 4, 1 to 6? I still don't really understand what that means.
Okay, can we wait? Because we are going to go over it. Okay, we're just jumping. I'm previewing a little bit. We will go over it. Okay, so polysaccharides are used for storage and for fuel, right? Storage, structural, and for fuel as they're broken down. Okay, polysaccharides, glycogen and starch. Right? Glycogen is how you store glucose in your body. In your muscles, you have glycogen granules, which are long chains of glucose put together. Okay? In plants, starch is long chains of glucose put together and stored for use as plants need them. Okay? And then some structural stuff, cellulose and chitin. All right? But the structure and function of a polysaccharide are determined by its sugar monomers, which monosaccharides are there, and how they're linked. We're going to really concentrate on how they're linked, because that's really what makes a big difference. So, starch, storage polysaccharide of plants, entirely glucose monomers. Okay? So it's glucose, 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 all the same. Plants store this as granules, all right? And the simplest form of starch is amylose. Have you heard about amylose before? Does that sound familiar to you? Yes? No? Maybe. How many of you took anatomy in high school? What is amylose? Or how do you know? Does that sound familiar to you at all? Alyssa?
It sounds familiar.
She doesn't remember. Okay, amylose is starch. Okay, and it's broken down begins to be broken down in the mouth by a enzyme. Oh, so like, I get by an enzyme called what? Amylase. Okay, so amylase is an enzyme. It's produced it's in the saliva by the salivary glands, and it will break down starch. It will begin to break down and take off bits of these glucose molecules. Okay, so amylase, the simplest form of starch. All right. We're going to go through the differences in these in a second. Glycogen is a storage polysaccharide in animals, right? And you store glycogen in your liver and in your muscles for quick production of glucose. Right? And again, it is all fuels. Okay? So, you see here's your starch granules. Okay? You see that starch, amylose, is a 1,4 linkage. Okay? Look at the difference between the linkage between starch and glycogen. Is there a difference? Can you see? Yes or no? Not really. Okay? There's not really a difference here. Right? Let's look at those two glucose right there as opposed to these two glucose molecules. It's not really a difference. Okay? And your body in amylase can recognize that linkage, okay, and break this starch linkage apart. Okay? But now look at the difference between starch and cellulose, which is another long chain of glucose molecules. Okay? This chain, it's attached to the 3 and 6, looks quite different than the other two, right? This glucose molecule right here has been flipped. Look where the oxygen is as opposed to where the oxygen is here. All right? It's on the opposite side. All right? So the oxygen molecules are on the opposite side. Now, what's the difference between starch and glycogen then? What do you see the differences between starch and glycogen? Plants and animals. Let's just go structure-wise. Starch is one chain. Okay? If you look at glycogen, it's branched. You see these? It has multiple chains. So glycogen, storage polysaccharide in animals, and is branched. Okay? While cellulose is not really branched, it is packed together, and there are linkages, or I wouldn't say linkages. There's chemical interaction between the strands. All right? You see these hydrogen bonds between the strands that help them pack together. And because the oxygen is flipped, right, in the wrong designation, we do not have an enzyme in our body that recognizes that structure. Therefore, cellulose that we consume, well, there's a text message. The cellulose that we consume passes through the body in the form of fiber. Right? It's not digestible. Right? However, other animals, cows, horse, whatever, those ruminants that consume cellulose can break it down. So to further explain the 1,4, 1,6 linkages, all right, and that's also one of the differences between starch and glycogen. Because of the 1,6 linkage, all right, it allows it to be branched. Okay? So if we were to draw a glucose molecule, and I just want to go over this so you understand this better. Right? We could, this being the oxygen, here's being carbon number 1, 2, 3, 4, 5, 6. Okay? And then, no, let's do this the other way. Let's number them the other way. 1, 2, 3, 4, 5, 6. Okay? Plus another one, glucose molecule, and we can put it together the same way, right? So this is 1, 2, 3, 4, 5, and then 6. Right? Now, if you're going to have a 1,4 linkage, it's going to link the oxygen between those two, right? You're going to end up with a certain structure. Now, if you put it on the 1,6, okay, on this 6 carbon way up here, right? There's a different structure that's going to be produced. With the different structure, it allows these other carbons, 1, 2, 3, 4, to also produce another glycosidic linkage. Am I confusing you more? Do you understand it better? You're looking at it like this. This is a messy drawing, I know. Okay?
We always number the carbons clockwise.
You don't have to worry about how you number the carbons now. Okay? For right now, the most important thing is to understand that there is the concept of the difference in linkages. I'm not going to ask you identify this as a 1,4 or a 1,6 linkage. Do you know that there's a difference between starch and glycogen? Yes? What is that difference in how those glucose molecules are arranged? Emily.
So we don't have to fully understand the difference. We just have to know that there is a difference.
I'm trying to show you the difference without getting into too much organic chemistry and breaking it down that way. Because in organic chemistry and biochemistry, you will do this. Okay? And one more time? Okay. All right, one more time. And I'll try to draw it better. Maybe. Let's delete that. All right. One more time. Let's draw the glucose. Let's put this down so I can draw it a little bit better. The glucose. Okay, with this carbon that's hanging off here, and that's an oxygen. All right, that's your glucose molecule, right? Well, if you number them, let's say clockwise, 3, 4, 5, 6. All right, one linkage can be between that first glucose, right? That's on one side, 1, through an oxygen to either the 4, right? If we number them the same way, or it could be done to the 6. Okay? Depending upon that structure, whether or not it is 1 to 4 or 1 to 6, gives this glucose here a different property. This 1,6 linkage allows for branching to occur. Okay? So you will find the 1,6 linkages in glycogen. You will not find the 1,6 linkages in starch or cellulose. Is that better? Do you understand that? It's a little more simple, better clarification? Yes? I see nods. I see nods. I see blank, understand, kind of nods. Okay. All right. Hopefully that's better. Can I move on past 1,4,1,6? Okay. Good?
What was cellulose?
Cellulose is 1,4, but it's but it's a flipped. So it's not cis, it's not what we call cis, it's called trans. All right, so cellulose is this. All right, so it's still one oxygen, this oxygen here, right, and oxygen there. The difference is for cellulose is where the oxygen is. So it's technically they actually write it like this as being the glycosidic linkage. Yep.
Is this your question?
Nope. She.
Okay. Is this the same as like if you have it on a straight line and then like the fourth one in is out there and the fifth one in? Or, do you know what I mean?
If you
Like,
have.
It was a straight line of like carbon.
Oh, if we're doing the linear structure.
Yeah.
No. The numbering, yes, 1, 4, if it's up like that, it's 4 up or 4 methyl, whatever it is, yes. But when we're talking about the numbering of carbon, we're talking about the number of carbon within the 6-ring structure. Okay, so we start at 1. So 1,4 linkage is the number 1 carbon on the first of the first molecule, and then the 4th carbon on the second molecule. All right? The other thing that this does, and I'm going to get rid of this again. The other thing that this does is allows you, so there's a 1, we'll call this a 1,4 linkage, right? That's a 1,4 linkage. Now, what can happen to this right here? What else is available now? This end carbon, or this end glucose can make another 1,4 linkage, okay, and continue to make 1,4 linkages. All right, giving your result in a long chain of glucose. Now the 1,6 linkage would then give you a branch, a branching. Okay, better understanding between that. So the main difference is starch is not branched, 1,4 linkages. Okay? Glycogen is branched, and because of the branching, you'll have 1,4, and where the branching occurs, 1,6 glycosidic linkages. All right? For cellulose, you have 1,4 linkages, but the second glucose is flipped. All right? So you don't recognize structure that way. Let's delete that. Let's bring this back up. All right. So the polysaccharide cellulose is a major component of cell walls. Right. Cellulose is a polymer of glucose glycosidic linkages, but those linkages differ. The 2-ring forms. So here, let's look at the 1,4 linkage versus the 1,6. All right, so here's your starch, 1,4 linkages of starch monomers, and you see the OH and the oxygen are on the same side. Okay, so for starch, you look at this oxygen, right, and the OH here. Okay, now that linkage, now the second glucose in that linkage is flipped. All right, the 1,4 linkage still the same between the same carbons, but it's going to be flipped on one end because where the oxygen and the hydrogen or the OH groups are, they're on opposite sides. Okay, because of where this group is and this OH group, that prevents our enzymes from recognizing cellulose as starch and breaking it down. All right, because of this difference in structure. Okay. Questions on cellulose versus starch and their structure. Okay. So in starch, the monomers are arranged in the alpha. We call the alpha conformation. All right. And then in cellulose, they're in the beta conformation. So it's the difference being an alpha 1,4 linkage in starch and a beta 1,4 linkage in cellulose. All right? So there, because of that, starch is largely what we call helical in shape, while cellulose is relatively straight. The straight nature of cellulose allows for the hydrogen bonding between glucose molecules and allows for strength, right, and support. So you have your hydrogen atoms of one strand, right, and then your parallel cellulose molecules are held together this way. And they're grouped into what we call microfibrils, and gives you your building material of plants. So your enzymes that digest starch, such as amylase, okay, they can't hydrolyze the different linkages, right? So an amylase enzyme will recognize the alpha, that's what this is, alpha linkages, and cannot hydrolyze the beta linkages. All right? In that case, cellulose in human food passes through the digestive system as fiber. Right? Some microbes, okay, will have and can recognize, have enzymes that recognize this linkage and will break it down and produce vitamins and so on off of that, and then you have your cows to termites have symbiotic relationships with these microbes in order to digest cellulose. Okay, and then the other one, the other polysaccharide, chitin, is another structural polysaccharide. It's found in the exoskeleton of arthropods. It provides structural support for cell walls of fungi. Okay, we are not going to be concerned with the structure of chitin. Just know that that is another polysaccharide, and it's used for structural support. You might find it in the exoskeleton of a lobster and other arthropods. Questions on carbohydrates, polysaccharides, monosaccharides, and their structure and function? No? Okay. So, with that, you are done for today.